I'm Madison Brown, and this is our muscle contraction project. Our first aspect is the muscle fiber. This is the muscle fiber, which is a muscle cell composed of myofibrils that help produce a contraction. These are the myofibrils, which are long protein cords filled with myofilaments that perform the muscle contraction. The myofilaments are in the myofibrils, which are actin and myosin that slide past each other to produce the muscle contraction. The sarcolemma is the cell membrane that acts as a barrier. The sarcoplasm is the cell's cytoplasm. The sarcoplasmic reticulum forms a network around each myofibril. The terminal cisternae are expanded chambers at the end of the sarcoplasmic reticulum that serve as a reservoir for calcium. The T-tubules are in between the terminal cisternae, which are extensions of the sarcolemma that are filled with extracellular fluid and form channels through the muscle fiber and release calcium bursts. Myofilaments make up the sarcomere, which is a contractile unit of the muscle that contains myosin and actin. The myosin are the thick filaments, and the actin are the thin filaments. The Z-disc separates each sarcomere and is the anchoring point for the actin myofilaments. The M-line is the anchoring point for the myosin myofilaments. The I-band is the light band and is the zone that just has actin and does not have myosin. The A-band is the dark band, and that is where the actin and myosin overlap. The H-zone is the area of only myosin. During contraction, the A-bands stay the same length, while the I-band and the H-zone shorten. The neuromuscular junction is on the muscle fiber, and it is a chemical synapse formed when a motor neuron and a muscle fiber make contact.
This is Lauren Bessler, and here's the process for a muscle contracting and then relaxing. First, an action potential is generated by the motor neuron, and an action potential reaches the axon terminal. Voltage-gated calcium channels open, and calcium enters the cell. Calcium signals the vesicles to bind to the neuron and the plasma membrane, and acetylcholine is released into the synapse. Once acetylcholine crosses the synapse and binds to the receptor of the sarcolemma, ion channels on the sarcolemma open, and sodium enters the cell. This causes the muscle cell to begin to depolarize. This local depolarization leads to an action potential propagation. The action potential travels along the sarcolemma to the T-tubule. Once at the triad, the action potential helps open voltage-gated calcium channels on the terminal cisternae. Calcium leaves the sarcoplasmic reticulum and enters the sarcoplasm. The calcium then binds to the troponin. Troponin changes shape, which moves the tropomyosin, uncovering the myosin binding sites on actin. The myosin then binds to the actin and the muscle contracts. Then the calcium is resequestered into the sarcoplasmic reticulum. Myosin releases actin. The troponin reconfigures. The sarcolemma repolarizes by closing sodium channels and allowing potassium to leave the cell. And finally, the muscle relaxes.
My name is Allison Carter, and I will be talking about how each sarcomere helps in the role of muscle contraction. So the first one is troponin-tropomyosin complex. Troponin is attached to the protein, and it alerts muscles that there is a structural change. Here's our troponin proteins. Tropomyosin lies between the actin filaments and muscle tissue. It's this yellow line here, as you see, and it blocks attachment sites for the myosin cross bridges, therefore not allowing contraction to happen. The next one is cross bridge formation. It is the attachment of myosin and actin in a muscle cell. The myosin is bound by ATP, which then becomes ADP from the ATPase. This then gives myosin a lot of potential energy. The myosin head then brings the gap to actin, forming a cross bridge. Here's our myosin, and as you can see, it attaches here to the actin, forming a bridge. After that, we have power stroke. This pulls actin toward the M-line. When actin is pulled, the sarcomere will shorten and the muscle will contract. It occurs when troponin takes ATP, cleaves it, and drops it, therefore allowing the power stroke to happen. As you can see, the head reaches up, grabs it, pulls it back, and then releases. Lastly, we have the myosin's head usage of ATP. The myosin's head pulls at the actin site, allowing for filaments to slide past each other. This allows for the cross-bridge cycle, and we also see that in this picture. Here is our myosin head with the ADP attaching to the active site right here. And this will allow the bridge cycle to continue to happen with this connection.
I'm Madison Liston. Myosin and actin are contractile proteins because they control the muscle's contraction and relaxation. They mediate the contractile fibers. Tropomin and tropomyosin are regulatory proteins because calcium binds to the tropomin, changing its shape. Tropomyosin, when it's relaxed, covers the myosin binding sites. These two result in the processes being turned on or turned off. Calcium is a regulatory ion because it regulates the channels involved in excitation. It binds to tropomin, regulating the myosin binding.
It is Allison again, and I will be talking about the 4 processes in a muscle contraction that use ATP and what would happen if ATP were not available. So the first is the sodium-potassium pump. If there was no ATP, there would be no gradient created. Therefore, sodium right here would not be able to be pushed in, and potassium would not be able to leave. Next, we have the calcium pump. If there is no ATP, calcium won't be pumped back into the terminal cisternae. Next, we have exocytosis. Without ATP, acetylcholine will not be pushed out because it cannot move against the gradient without ATP. And lastly, we have tropomyosin uncovering actin bindings. If there is no ATP, cross bridges would not be formed because the tropomyosin would not be able to pull back the coverings from the actin.
It's Lauren, and I'm going to be talking about what happens to muscle contraction if a person is anemic. A common symptom of anemia is muscle weakness, and this is because the muscle does not have enough energy to hold a muscle contraction. Anemia is caused by a lack of red blood cells, which you can see in this picture, and red blood cells are used to carry the oxygen to muscles during exercise. If there is not enough oxygen, then the muscle will lose valuable ATP and then lose its ability to function and contract.
I'm Madison Liston, and I'm going to be talking about what happens to the muscle contraction when there's dehydration. Dehydration is when there's not enough water present in the body and it causes an electrolyte imbalance. This results in involuntary muscle contractions and muscle cramping.